As I mentioned in my previous video on photosynthesis, plants convert light energy into high-energy compounds ATP and NADPH through the light reaction. ATP and NADPH are then used to fix carbon dioxide as glycerolaldehyde 3-phosphate, abbreviated as GA3P, during the Calvin cycle. Glycerolaldehyde 3-phosphate is an intermediate of glycolysis and can be further oxidized to generate energy. Plants store extra carbohydrate as starch and sucrose. Starch is a homopolysaccharide made of glucose residues. It contains the linear component known as amylose linked by alpha-1 to 4 glycosidic bonds, and the branch component known as amylopectin linked by alpha-1 to 4 as well as alpha-1 to 6 glycosidic bonds. Starch biosynthesis occurs in the stroma of chloroplast. First, glucose 6-phosphate, abbreviated as G6P, derived from 2 molecules of glycerolaldehyde 3-phosphate through gluconeogenesis, is converted to glucose 1-phosphate, or G1P, by phosphoglucomutase, abbreviated as PGM. ADP glucose pyrophosphorylase then condenses glucose 1-phosphate with ATP to form ADP glucose, an inorganic pyrophosphate, which is cleaved by pyrophosphatase into 2 molecules of inorganic phosphate. Starch synthase then transfers glucose residues from ADP glucose to a non-reducing end of pre-existing starch molecules, releasing ADP molecule. Similar to glycogen synthesis, chloroplasts also contain a branching enzyme that introduces alpha-1 to 6 branches, producing amylopectin of starch. Sucrose is a disaccharide made of glucose and fructose. Sucrose biosynthesis occurs in the cytosol. Glycerolaldehyde 3-phosphate from the Calvin cycle is first converted to dihydroxyacetone phosphate, abbreviated as DHAP, by triose phosphate mutase, abbreviated as TPM. Dihydroxyacetone phosphate is then exported out of the chloroplast through an antiporter that simultaneously transports inorganic phosphate into the chloroplast for ATP synthesis during photosynthesis. Once exported to the cytosol, dihydroxyacetone phosphate is reconverted to glycerolaldehyde 3-phosphate. Similar to gluconeogenesis, aldolase condenses glycerol 3-phosphate with dihydroxyacetone phosphate to form fructose 1,6-bisphosphate, abbreviated as F16BP. Fructose bisphosphatase 1 then hydrolyzes fructose 1,6-bisphosphate into fructose 6-phosphate, which is one of the precursors of sucrose biosynthesis. Fructose 6-phosphate can be isomerized by phosphohexyl isomerase to form glucose 6-phosphate, which can then be converted to glucose 1-phosphate by phosphoglucomutase, similar to starch biosynthesis. Next, UDP glucose pyrophosphorylase condenses glucose 1-phosphate with UTP to form UDP glucose and inorganic pyrophosphate. Sucrose 6-phosphate synthase then condenses UDP glucose with fructose 6-phosphate to produce sucrose 6-phosphate, releasing UDP. Finally, sucrose 6-phosphatase hydrolyzes sucrose 6-phosphate into sucrose, releasing an inorganic phosphate. Starch and sucrose biosynthesis must be coordinated with the rate of photosynthesis. During the day, when light reaction is active, more ATP and NADPH is produced, fueling the Calvin cycle to synthesize abundant 3-phosphoglycerate, abbreviated as 3PG, that activates ADP glucose pyrophosphorylase, leading to increased starch biosynthesis. During the night, when photosynthesis ceases, inorganic phosphate builds up as ATP is not synthesized in the light reaction. Inorganic phosphate inhibits ADP glucose pyrophosphorylase, decreasing starch biosynthesis. Since sucrose is made of both glucose and fructose, sucrose biosynthesis is regulated by the availability of both substrates. The availability of fructose 6-phosphate is regulated by the activity of fructose 1,6-bisphosphatase 1, abbreviated as FBPase1, as well as the enzyme that reverses its action, pyrophosphate-dependent phosphofructose kinase, abbreviated as PPPFK1. Similar to the coordinated and reciprocal regulation of glycolysis and gluconeogenesis, FBPase1 and PPPFK1 are also allosterically regulated by the side product fructose 2,6-bisphosphate, which is produced from fructose 6-phosphate by phosphofructose kinase 2, abbreviated as PFK2. Fructose 2,6-bisphosphate can be reconverted back to fructose 6-phosphate by fructose bisphosphatase 2. During the day, 3-phosphoglycerate and dihydroxyacetone phosphate produced by the Calvin cycle inhibits PFK2, reducing the level of fructose 2,6-bisphosphate, whereas during the night, inorganic phosphate activates PFK2. increasing the amount of fructose 2,6-bisphosphate, which activates pyrophosphate-dependent PFK1 while inhibiting FBPase1, thereby decreasing the amount of fructose 6-phosphate and decreasing sucrose biosynthesis. During the day, PFK2 is inhibited, decreasing the level of fructose 2,6-bisphosphate, preventing it from inactivating FBPase1, thereby increasing fructose 6-phosphate and sucrose biosynthesis. On the other hand, sucrose 6-phosphate synthase is regulated by phosphorylation. It is active while dephosphorylated and inactive while phosphorylated. Sucrose 6-phosphate synthase kinase, which phosphorylates sucrose 6-phosphate synthase, is allosterically inhibited by glucose 6-phosphate, while sucrose 6-phosphate synthase phosphatase, which dephosphorylates sucrose 6-phosphate, is allosterically inhibited by inorganic phosphate, which means that ultimately during the day, excess glucose 6-phosphate indirectly activates sucrose 6-phosphate synthase, promoting sucrose biosynthesis. And during the night, accumulation of inorganic phosphate indirectly inhibits sucrose 6-phosphate synthase. Plant cell wall consists of homopolysaccharide of glucose known as cellulose. Cellulose biosynthesis is facilitated by rose-shaped complexes located on the plasma membrane, which is one of the shared derived traits of land plants and the green algae chaerophytes. First, glucose is attached to a plant sterol cytosterol on the inner surface of the plasma membrane. The intracellular cellulose synthase adds more glucose residues to the first one, in beta 1-4 linkage, forming a short oligosaccharide chain attached to the cytosterol, known as cytosteroldextrin, which flips across the outer face of the plasma membrane to serve as a precursor of cellulose. Next, sucrose synthase converts sucrose and UDP to UDP glucose and fructose. Cellulose synthase then transfers glucose residues from the cytosolic UDP glucose to elongated cellulose. Sucrose synthase and cellulose synthase forms a rose-shaped complex that directly feeds UDP glucose from sucrose into cell wall biosynthesis. Each 6 globules of the complex synthesizes a cellulose chain. The enzyme complex moves along the plasma membrane following the course of microtubules in the cytoplasmic layer just under the membrane. Parallel cellulose chains are eventually crystallized to form cell wall fibrils. Many bacterial cell walls consist of the heteropolysaccharide peptidoglycan, which is made of repeating units of N-acetylglycosamine, abbreviated as GlucNAc, and N-acetylmuramic acid, abbreviated as MER2Ac, linked by beta 1-4 glycosidic bonds. First, GlucNAc 1-phosphate condenses with UTP to form UDP GlucNAc, which reacts with phosphoenol pyruvate or PEP to form UDP MER2Ac, coupled to the oxidation of NADPH to NADP+. Next, MER2Ac combines with 5 amino acids and the combined moiety is transferred from UDP to the membrane lipid dolichol, a long-chain isoprenoid alcohol. Next, GlucNAc is also transferred from UDP to the dolichol complex. 5 additional glycine residues are attached to the lysine residue of the pentapeptide, forming a disaccharide decapeptide, which is added to the non-reducing end of an existing peptidoglycan primer. Finally, a transpeptidation reaction between the peptide chains on 2 different peptidoglycan molecules form a crosslink that contributes to a huge, strong bacterial cell wall. The antibiotic penicillin inhibits the transpeptidation reaction, thus killing bacteria by weakening their cell walls.